All right, so I'd like to thank you for the opportunity to speak here today about some of the work that my laboratory does on cell migration and human natural killer cell development. So as I said, my lab studies human NK cell development, and we do this in a number of different ways. One of the ways that I'm not going to talk about today is through the study of patients with rare primary immune deficiencies that affect NK cell development, and as a result have NK cells that either aren't present or don't work. What I am going to talk more about today is some of the work that we do on the cell biological aspects of human NK cell development. So specifically understanding how human natural killer cells and other innate lymphoid cell subsets develop within tissue and the signals that they have received from their microenvironment that facilitate their differentiation. And then I'm also going to touch upon some work that I have done and continue to do on the quantitative analysis of the natural killer cell immunological synapse. So what is an NK cell? So NK cells are non-T, non-B lymphocytes of the innate immune system. So by definition, they lack antigen-specific receptor rearrangement. They're derived from the common lymphoid progenitor, and they're most closely related to the other innate lymphoid cell subsets, so specifically ILC1, ILC2s, and ILC3s. They circulate within peripheral blood. They also reside within tissue and exert important functions there, primarily through the secretion of cytokines. And tissues where we know that they're important include uterus, liver, skin, and lung, as well as secondary lymphoid tissue. And we know in part through our study of these patients with primary immune deficiencies that affect NK cell development or their function, that they're critical for the control of viral infection, particularly those of the herpes virus family members. So specifically varicella-zoster virus, Epstein-Barr virus, and cytomegalovirus. And they exert this function in part through the contact-dependent lysis of either virally infected or malignant cells. So similar to a CD8 positive effector T cell, they form an immunological synapse, and they utilize the directed secretion of perforin and granzyme containing lytic granules towards target cells to kill them. And then they also play an important role in the production of cytokines. So although they're most closely related to the innate lymphoid cells, functionally they align quite closely with a CD8 positive effector T cell. So when we consider NK cell lytic function, so this directed secretion of lytic granules, this is performed through the formation of an immunological synapse, a lytic immunological synapse. And we can think of this broadly in a number of different stages. Now, each of these stages contain molecular events that help to regulate this function and dictate this function. But generally speaking, the way that I tend to think about this is through an initiation stage, an effector stage, and a termination stage. In the initiation stage, we see the first contact between an NK cell and a prospective target cell. This is mediated primarily by integrins, and then upon signaling either through activating or inhibitory receptors, the NK cell either decides to commit to killing the target cell or disengages and moves on. So with sufficient activating receptor activation signaling, we see the firm adhesion of the NK cell to the target cell. And so this includes further adhesion mediated again by integrins. We see actin remodeling, which helps to stabilize this adhesion as well as dictate receptor movement at the synapse to further amplify activation signals. And then an early step in NK cell immune synapse formation is the movement of these lytic granules, which contain perforin and granzymes, along the microtubules to the microtubule organizing center. So we see the convergence or the coalescence of all these lytic granules around the MTOC. In the effector stage, we see the polarization of the natural killer cell towards its target. And in this case, we see the fairly massive polarization of the MTOC along with the lytic granules and a number of other elements of the lytic machinery towards the target cell. So we see the NK cell polarized towards the target. We see the lytic granules along with the MTOC come to the synapse. And then in the degranulation stage, we see these lytic granules traverse the cortical actin that's found at the synapse and undergo exocytosis in order to initiate apoptosis within the target cell. And following this, we get detachment of the NK cell from the target, at which point they can move on and go engage with additional targets. And so shown here on the left is an example of a fixed cell confocal image of an NK cell shown here on the top, about to kill a susceptible target cell shown here on the bottom. And so we can see that the lytic granules containing perforin are stained in blue, and we can see that they've converged around the microtubule organizing center, which is shown here stained in red. And then this has been polarized at the immune synapse. We can see that the F-actin in the NK cell has remodeled to enable the firm adhesion to the target cell. And we can see that the lytic granules are poised about to undergo exocytosis onto the target. So we can even see that there's a lytic granule that's positioned underneath the MTOC, and another one here that's actually sort of embedded in the actin, ready to travel across the immune synapse and initiate apoptosis in the target cell. Now, when we consider human natural killer cells, obviously the easiest place to access these is within peripheral blood. And when we look within peripheral blood, we classify human NK cells primarily by the expression of CD56 and the absence of the expression of CD3. So we consider them to be CD56 positive, CD3 negative, which distinguishes them from NKT cells, which are CD3 positive. Now, when we look at these CD56 positive, CD3 negative cells in peripheral blood, we can further classify them based on the density of the expression of CD56 itself on the cell surface. And so here we can see that the majority of cells within peripheral blood in a healthy adult human are CD56 dim, so they have a lower density of expression of CD56. And this is approximately between 80 and 90% of the NK cells that you find within your peripheral blood are the CD56 dim phenotype, whereas the minority of these cells are CD56 bright, which again is marked by this much higher density of CD56 expression on the cell surface. Now, when we look more closely at these subsets, we can see that they're not just delineated by the expression of CD56, but these actually represent two specialized functional and phenotypic subsets of NK cells found within peripheral blood. So when we first look at the CD56 bright subset, which is of course the minority, we can see that it is marked, as the name implies, by the increased density of CD56 itself. These cells also express markers that are associated with immaturity and with tissue residency. So this includes CD117, which is c-KIT. They express the high affinity IL-2 receptor, which is shown here. They express CD62 ligand and CCR7, which again are associated with tissue residency or homing, as well as CD127, which is the IL-7 receptor. And they express transcription factors, including EOMES and TBET. And these cells are very potent producers of cytokines with minimal further activation, so particularly interferon gamma and TNF alpha. In comparison, when we look at the CD56 dim subset, this is the majority of cells in peripheral blood, again marked by the lower density of CD56. And these cells also express other markers that are associated with NK cell maturation. So these include the KIRs, or killer immunoglobulin-like receptors. They express CD16, which is the FC receptor, which enables these cells to perform antibody-dependent cellular cytotoxicity. And then they also express other markers that are associated with activation for cytotoxicity, so the natural cytotoxicity receptors and NKG2D. And they have slightly higher expression of TBET and slightly lower expression of EOMES than the CD56 bright cells. And these cells are generally thought of being the primary cytotoxic cells at baseline. So they express perforin and granzyme at baseline and are less inclined to produce cytokines, although it should be noted that either of these subsets, if you activate them in the right way, will perform all functions that an NK cell will perform, so either cytokine production or cytotoxicity. Now, the relationship between these two subsets that we find within peripheral blood is thought to represent the terminal stages of NK cell maturation, which is a process that begins in the bone marrow with a hematopoietic precursor. And then one of the things about NK cell development, particularly relative to T cells and B cells, is that we really don't fully understand, how and where this process takes place. So we know that the precursors arise in the bone marrow, and this is a necessary source for these cells. We think that these cells at some point then exit the bone marrow, enter circulation, and then seed peripheral tissue in order to undergo further development. Now, the best characterized site of this is secondary lymphoid tissue. And from secondary lymphoid tissue, we can isolate the spectrum of cells which we think represents the spectrum of NK cell development. And we can classify these both based on the expression of cell surface markers and other phenotypic signifiers, and also by their increasingly restricted lineage commitment. So at each of these stages, you lose some of the potential to become other cell types. Now, that being said, there's a fair amount of plasticity. So for example, at the stage 3 stage, these cells still have the capability or the potential to make other innate lymphoid cells. But by the stage 4A and 4B, we're really committed to an NK cell lineage. So as I mentioned, the best characterized site of this is secondary lymphoid tissue. But that being said, I think it's certainly likely and in fact probable that this can be carried out in other tissue sites as well, particularly those sites that have specialized NK cell subsets, as I mentioned, like uterus and liver. In addition to which, you can also isolate the spectrum of NK cell development from bone marrow, and so it's likely that this process can also take place there. However, whether these precursors enter circulation and then return to bone marrow to seed, or whether they just stay there is unknown. So again, when we look within peripheral blood, the stages that we find there are thought to be representative of stage 4 and stage 5, or which is analogous to the CD56 bright and CD56 dim subsets. And then within peripheral blood, we also find what we think of as a terminally mature subset, which is CD56 dim cells that express CD57. Now, we can also recapitulate this in vitro, and when we do that, we take CD34 positive precursors from a variety of sources, either from induced pluripotent stem cells or embryonic stem cells, or from cord blood, or from peripheral blood, and we co-culture these with stromal cells in the presence of cytokines. And when we do that, these cells progress through development in a very similar way to what we find in tissue. So it's a process that takes about 4 weeks. And we can see them, we can track their progress through the stages of NK cell development as they're listed here. And the stromal cell lines that are frequently used for this are commonly used for other programs of lymphocyte development. So they include things like the OP9 and EL08-1D2 stromal cells. So when I think about NK cell development and cell migration, I kind of think about this in 2 different ways. One is the migratory behavior that occurs in complex environments. So this includes trafficking to and from sites of infection through circulation and tissue. I think about this a lot in the context of what I just talked about with a precursor that you may exit the bone marrow and then seed peripheral tissue. How are those cells directed to where they're going, or is that a stochastic event? And of course, this includes, is going to be strongly regulated by processes that include chemotaxis, as well as durotaxis and haptotaxis, which of course occur in response to signals from the microenvironment. And so shown here is just a schematic of what we think might be mediating the recruitment of one of these precursors from circulation into tissue where they can undergo development. Although of course we have a very poorly understood idea of how this is being mediated. And then the other way that we think about this is something that my lab has identified, which is really the spontaneous migration of NK cells as they're undergoing differentiation. So on the previous slide where I outlined NK cell development, one of the things that I had highlighted down at the bottom was this increasing polarization and cell migration phenotype. And this is something that we kind of unexpectedly found when we were using some of these in vitro models to generate NK cells, generate human NK cells. And what we found was that when we first plated these cells as CD34 positive precursors, they were largely non-polarized and non-motile. And then within a fairly short amount of time on these stromal cells, we would see the cells begin to migrate. And they would migrate seemingly almost continuously for the entire time of culture, which is, again, between 21 and 28 days. And as they progress through differentiation, we see increasing complexities to these behaviors. And so we can break these modes of migration down into their individual components. And these include things like directed migration, non-directed migration, and arrest. And we see this sort of fairly characteristic sequence of changes that occurs as they go through development. So we initially thought that this was something that was, probably an artifact from an in vitro culture system. But what we further found, and I'm not going to talk too much about this because it's been published, but what we found was that if we isolate NK cells from tissue where they're undergoing development, we see that these characteristics are associated with the developmental stages that we find from freshly isolated cells as well. And so shown on the left is a cell migrating. So what we've done here is we've laid down a sort of a carpet of stromal cells. The NK cell, the developing NK cell is shown labeled by CD56 itself in red and CD62 ligand in green. And you can just see that these cells are undergoing fairly significant migration on the stromal cells as they're undergoing differentiation. And the graphs down here also illustrate this in a slightly different way. This is just tracking these cells at individual time points as they're going through differentiation. So we can see at the earliest time point when we first seeded them, they're largely non-motile. Within 7 days, and particularly by 14 days, we start to see this sort of significant displacement from the point of origin in the tracks as we've tracked a number of cells. And then we can also measure this in terms of the complexity of these behaviors by the number of times that they switch between modes of migration. So we can see at the earliest time point, they're primarily in one mode of migration, and that mode is non-motile essentially. And then progressively they begin to have more complex behavior, and at the latest time point, we see them switching frequently between these different modes of migration, either directed, non-directed, or under arrest. So when we look more closely at cell migration itself, lymphocyte migration is, of course, as with all cell migration, is a cyclical event that requires the polymerization of actin. So we see the protrusion of an actin leading edge. We see adhesion of the cell through the mid-body, and then we see retraction of the uropod. And so all of these events are required for the cells to migrate. So in this case, we can think of the cell going in this way. And we can see this when we look at an example of a migrating cell, which is shown here on the left. This is an NK cell going in this direction. And so we can see this this beautiful sort of branched actin network as the lamellipodia. We see additional cortical actin, which is less dense, but still branched within the cell body. And then we see the trailing uropod. And so, of course, this is a process that's heavily dependent on actin remodeling. So for the formation of this lamellipodia, we can see that it's branched actin networks that are formed by ARP2/3 and other actin regulators. And it's also heavily dependent on integrins, which help to mediate traction, particularly through the mid-body of the cell. And in addition, help to mediate adhesion to the extracellular matrix. And of course, NK cells, like all lymphocytes, are quite rapid in their migration. And so we also know that the contraction of the uropod is really important for this process. And we can see that NK cells, as shown here in this confocal image down at the bottom, and this has been described before, actually will elevate their uropod in order to be able to move faster, or it's thought to be able to move faster. And so we can see this when we're looking from the side into the Z profile here. We can see that the uropod, which is marked in green, is elevated as the cell's migrating in this direction. So the work that I'm going to talk about today is unpublished work that is really focused on trying to understand better one of the sort of the unexpected things that we identified when we were classifying some of these modes of migration on these stromal cells. And this is work that's been done primarily by a student who was first an undergraduate in the lab and is now a postbac researcher, Mara Dixon. And this is a study of CD56 itself, so the canonical marker of human NK cells, and the role that we think that it's playing in cell migration. So again, I've already introduced you to CD56. It's expressed highly, particularly on the CD56 bright subsets, less so on the CD56 dim subsets. It's part of the Ig superfamily and contains an extracellular domain with fibronectin domains and Ig repeats. And then it has a transmembrane domain and an intracellular domain. And there are 3 isoforms of CD56 because CD56 is actually NCAM, so a neural cell adhesion molecule, and it can be expressed as either a GPI anchored 120 kilodalton form, or the form that we think is the primary isoform found in NK cells, which is NCAM 140. Now, we don't know a lot about the function of CD56 or NCAM on NK cells, but we do know a little bit more about its function as a neural cell adhesion molecule. And so we know that it can undergo direct signaling through focal adhesion kinase and through the Sark family kinase FIN, and this leads to neurite outgrowth. It can also bind to FGFR1, both in cis and in trans, to help promote cell migration. And then it can undergo homotypic binding, both in cis and in trans, to mediate adhesion. And then finally, although it's less well understood, it can undergo ectodomain shedding, which helps to activate beta-1 integrins. So when we look at the expression of CD56 on human NK cells, and in this case, we're looking by STED microscopy, and we've activated the cells on glass, and then we're looking back up into the NK cell. So this is looking right at the plane of the glass into the cell. And we can see that, consistent with it being a cell membrane receptor, CD56 is found associated with, sort of the cortex of the cell, or with the actin cortex. And so we can see, this branched active network. We see CD56 out on the membrane. We also see it very prominently decorating philopodia. So when we look at these extensions that are coming out from the NK cell here, we can see the presence of CD56, particularly on the philopodia, decorating these. And then notably, we also see it at the point of contact between NK cells and other NK cells, and that's just highlighted here as well. So of course, we're not the first people to wonder what the functional role of CD56 might be on human NK cells, and there was a lot of work that was done, particularly when CD56 was identified as NCAM. But a lot of that work really was focused on the homotypic interactions that it can undergo in order to mediate adhesion. So it was determined fairly early on that if you block CD56 interactions between NK cells and target cells that express CD56, you don't see an effect on tumor target cell lysis. So it doesn't seem to be mediating adhesion, at least in the context of cytotoxicity, by binding to other CD56 molecules on the target. And we do know that CD56 can bind to FGFR1 on T cells and stimulate T cell production. So this is a productive interaction that occurs between NK cells and T cells that's mediated by CD56. And then it also has a role, it seems, in binding to fungus, so particularly Aspergillus, and it can promote NK cell chemokine secretion that way. Now, in work that we published last year, and again, I'm not going to talk too much about this because it is published, but we fairly unexpectedly found a role for CD56 in regulating cytotoxicity with NCAM, or with CD56 negative targets. And so this was work that was done by a graduate student in the lab, Justin Gunnish, in which we knocked out CD56 on the NK92 cell line, and again, somewhat surprisingly, found that this severely impaired their cytotoxic function. When we put CD56 back in, it was restored, suggesting that this is specific. And without getting into too many of the details, what we found was that this seemed to be regulated through PIK2, or protein tyrosine kinase 2. And so what we found was that in the CD56 knockout cells, we found decreased phosphorylation of PIK2, again, which was restored upon the re-expression of CD56. And we tested this with a couple different NK cell lines. We found that CD56 was important for cytotoxicity in the NK92 line, but not in the YTS line. And then we think this is related to the usage of PIK2 by these two cell lines for cytotoxicity. So one of the reasons that we had focused on PIK2, or that we became interested in PIK2 in this study, was because of the previously described role that it and focal adhesion kinase can play in regulating NCAM in other settings. So as I mentioned, NCAM 140 can bind to FIN, which is a Src family kinase, which is expressed in neuronal cells and interestingly is also expressed in NK cells. And this recruits focal adhesion kinase to initiate growth cone migration. Now, focal adhesion kinase is a homologue of PIK2. So PIK2 is a lymphocyte-specific focal adhesion kinase. And it's previously been shown that PIK2 is required for NK cell cytotoxicity and immunological synapse function. And then PIK2 also contains multiple phosphorylation sites, and these include tyrosine 402, which is the one that we were measuring in the graph I just showed you. And this is the initial site that's required for autophosphorylation and then subsequent activation, as well as tyrosine 881, which is interesting because in CTLs, or cytotoxic T lymphocytes, this actually helps to promote uropod detachment. So, PIK2 is it's in addition to these functions, it's downstream of integrins. It functions downstream of integrins for initiating integrin signaling, and so it seems to be required for both sort of initiating actin remodeling, as well as helping to break down focal adhesions or integrin-associated complexes in order to help cells detach. So our interest in CD56 actually began with this initial paper where we were looking at the migration of cells on stromal cells, where we found that if we blocked CD56 or we knocked it out in primary cells, we impaired the cell migration that I showed you, the spontaneous cell migration of these cells on the stromal cells that we were using. Now, the first thing we thought about was whether there were homotypic interactions that were occurring between CD56 on the NK cells and on the stromal cells. And we find that the commonly used stromal cells that we use for NK cell development, so both the OP9 and the EL08-1D2 cells, do express NCAM, so that's shown here in magenta. So we think this may still be playing a role, and that is work that, again, I'm not going to talk about today, but is ongoing in the lab. But what we wanted to focus on was independent of that, because we found that when we knocked out CD56, either in the NK92 or in the YTS cell line, we found impaired, similar to what we had found with the primary cells, we found impaired migration. And this was both on stromal cells as well as on other substrates, including other integrin substrates such as fibronectin. And so what we can see here in the graphs on the right, which kind of look like silly string, are in the wild type conditions where we've tracked individual cells over time. We can see sort of significant displacement from the point of origin of these tracks and their velocity, whereas in the knockout cells we see minimal migration on fibronectin in this case. Let's see if these movies will play. There we go. And so another way that we looked at this was to use interference reflection microscopy. So in this case, we're putting down fibronectin on the glass, and then we're imaging the cells almost from the bottom, so looking up into the cell, almost looking at their shadow. And so as you can see from the CD56 knockout cells, when we drop them onto fibronectin, the cells minimally migrate. So it almost looked like the cell was stuck. Whereas when we look in the wild type condition shown here on the left, we can see that the cell just migrates through the frame of view. So this really kind of alerted us to the fact that the CD56 knockout cells were seemingly unable to undergo migration on fibronectin. We can see this a little better here when we look at stills from these movies. And in this case, what Amara's done is she's, again, coated fibronectin and then just dropped either wild type or CD56 knockout cells down onto the glass and then imaged them using interference reflection microscopy. And so we can see that the wild type cell hits the glass, kind of polarizes in the direction that it's going to go. So we see the formation of a leading edge, and kind of the contraction of its uropod, and then the cell migrates off into sort of out of the frame. Whereas in the case of the CD56 knockout cell, again, we see that we drop the cell down. It actually seems to try to polarize, so it appears to try to form a lamellipodia or a leading edge, but it appears like the uropod is stuck, and so the cell kind of tries for a couple frames and then actually gives up and kind of rounds up. Now, when we look more closely at this using structured illumination, we can see that these cells, the CD56 knockout cells, really are quite messed up. So again, on the left, we see this wild type cell, which is polarized with a lamellipodia, a nice leading edge, and a trailing uropod. And we can see a few filopodia, particularly at a region where the cell is starting to extend another little actin protrusion here. Whereas when we look at the CD56 knockout cells, we can see that they they appear to lack direction. So we don't see the formation of a nice lamellipodia, and that's one of the features that we've highlighted here. We also see the presence of these large actin foci within the cell body. And then, of course, probably one of the most striking things are these long dysregulated filopodia that look kind of long, branched, and somewhat angry. Now, this was reminiscent of what we had seen when we were initially studying these cells in the context of their cytotoxic defect. So when we were looking at the role that CD56 might be playing in cytotoxicity, we had done similar studies where we'd activated cells on glass. So in this case, we're using recombinant ICAM as well as anti-NKP30. So this provides a signal to the NK cell that recapitulates the immune synapse. And then again, we're looking up into the plane of the glass, in this case, staining with colloidin. And when we did this with either wild type or CD56 knockout NK cells, what we found was that similarly to what I just showed you with the migrating cells, we found kind of three hallmarks of the CD56 knockout cells. So one is the lack of a lamellipodia. So again, shown here, we see this isn't present in the CD56 knockout cells. The second, and perhaps the most striking in this context, is these large actin foci, which are highlighted here. And then the third are these longer filopodia that we find in the knockout relative to the wild type cells. And these actin foci are just quantified here on the right, where we see the higher frequency of the larger foci within the CD56 knockout cells. Now, the filopodia defect, I think, is interesting. And again, I'm not going to talk too much about this today, but one of the things I just want to highlight is that we actually see this defect in the absence of activation. So both the slides that I showed you previously are cells that have been put onto a surface that's going to either engage integrins and or activating receptors. But we actually see the filopodia defect at baseline. So essentially, if we just pull these cells out of culture and put them on an inert surface, we can see that, again, just qualitatively, there's a difference between the wild type and the knockout cells in terms of the length of their filopodia. But what I want to focus on a little bit more is this actin phenotype, and particularly these large actin asters that we find. And again, we find them in the context of activating in immune synapse recapitulation, so activating on glass with ICAM and activating receptor, or in the cells that are trying to migrate on other integrin substrates. And so when we look more closely at these using 3D structured illumination microscopy, what we can see is that they're both bigger in the XY plane. So we see these large foci. And then when we look up into the cell from the Z perspective, we can see that they're taller as well. So they seem to be these large, dense accumulation of actin within the cortical actin network. So one of the things we started thinking about, particularly as we knew that there was this role for PIK2 in CD56 mediated function, as well as an integrin function, was whether these represented integrin associated complexes. So of course, integrins link the cell membrane to actin through a number of different adapters. These include proteins such as vinculin and talin, as well as, of course, other adapters and regulators, including PIK2 itself. So when we look more closely at these, we can find that what was actually quite striking was the increased frequency of vinculin in these complexes. So again, we're looking at a wild type cell shown here on the top and a CD56 knockout cell. So we can see on the top here in the wild type cells, sort of smaller actin foci, and in the CD56 knockout cells, again, we see these large actin foci. And what's quite striking is that, one of the things about lymphocytes is that they don't tend to make very large integrin-associated complexes. So as you can see in the wild type cell, we don't actually see a lot of vinculin within the actin cortex of this NK92 cell. But within the knockout cell, we can see very prominently the association of vinculin, the increased density of vinculin, and its association with these actin asters that we find in the cortex. And we've quantified this here on the right, both in the actin foci, as well as within the lamellipodia and the filopodia. So we then wanted to look more closely look more closely at PIK2 itself. So as I mentioned previously, we had defined that decreased phosphorylation of PIK2 was associated with this decreased cytotoxicity in the CD56 knockout NK92 cells. And so again, we've activated the cells on glass here, and then we're looking up into the cell. And in this case, we've stained for the tyrosine 402 specific form of phospho-PIK2. So what was interesting was that when we looked more closely at this phospho-PIK2 defect, we could see that there was, in fact, decreased PIK2 at the synapse, but it was associated with these larger actin foci. So we actually were finding that although there was sort of less staining for PIK2, it was found with higher intensity and in seemingly larger foci in the CD56 knockout cells. And so we can see this here when we take a close-up of the cortex and look at the intensity of phospho-PIK2 staining. So it almost appeared like There was less PIK2 staining, but it was more concentrated into these larger foci itself. When we quantify this, this is what this demonstrates to us. So first of all, we found that we did find a slightly decreased cortex area in the CD56 knockout cells, but we find a significantly decreased number of foci that we detect of phospho-PIK2 in the CD56 knockout cells compared to the wild type. And we can see this also when we just look at the distribution of the size of these. So first of all, of course, in the wild type cells, we detect a higher number of the phospho-PIK2 foci when compared to the knockout cells. And in the knockout cells, these are more skewed towards the larger distribution. And then we find a significantly higher intensity of phospho-PIK2 staining itself within the CD56 knockout condition. And then finally, we wanted to look at the other tyrosine that is, of course, of interest to us in PIK2, which is this tyrosine 881. And this is the one that is also thought to be at least partially responsible for uropod detachment in CTLs. And we found a very similar pattern of staining, where we find that there is seemingly less phospho-PIK2 881 in the CD56 knockout cells, but that it's highly associated with the F-actin that's found in these large asters in the CD56 knockout cells relative to the wild type cells. So in summary, we still don't fully understand what's going on, but I think we're thinking about this in kind of one of two models. So one model may be that PIK2 is either directly or indirectly, and this may be related to FIN binding to CD56. And helping to mediate some function that way, either in concert with integrins or independently of them. And then one of the things we also think about is the fact that CD56 is a fairly large protein, and it's also modified by the addition of polysialic acid. So one of the things we wonder is whether it's also maybe playing a role in helping to regulate sort of the spatial modulation of integrins on the cell surface, in the sense that when you delete it, we get this increased clustering of integrins, which could then lead to some of these seemingly larger integrin-associated complexes that we're finding in the cortex. And then again, while I didn't talk about it today, I still do think that there could be a role for CD56 NCAM interactions, so homotypic interactions between NK cells and the stroma in particular, and we're also investigating that. And so with that, I'd like to thank my lab. As I said, I think all of the work that I showed you here today was done by Merritt Dixon, who's a very talented now post-bac researcher in the group. And I would also like to thank Justin Gunnish, who's not shown, but was the PhD student who did the cytotoxicity work. And thank you for attending.